This slide or presentation is on the adaptive immunity, which is the specific defense or that third line of defense. This is just a short PowerPoint just to summarize everything, hopefully putting everything in perspective and putting all the pieces together. Remember, it does involve the B cells, which are part of the humoral immunity. The main function is to produce those antibodies that are going to bind to those antigen, the foreign substance. This will help to inactivate it, and also it's going to flag it or target for destruction. It's also going to produce memory cells that in any subsequent future encounter with that antigen, it will trigger an immediate. The T cells are part of the cell-mediated immunity. There's several different types of T cells, your helper T cells, regulatory T cells, cytotoxic T cells, to name some examples. The main function of cell-mediated immunity is to help other immune cells. It's to help suppress or kill any abnormal cells like cancer cells. It is going to trigger the production of cytokines. These are chemicals that are going to be involved with helping to oftentimes stimulate other macrophages, stimulate cells to be able to recognize certain markers that target cells for destruction, etc. And it's also involved with some of the hypersensitivity. This slide is showing the clonal selection and differentiation of the B cells. And just something that I want to point out, because we've already gone over this, that the antigens have been presented. You start the clonal selection process, and some of these B cells will become memory cells. Others are used right away to produce the plasma cells, which in turn produce the antibodies, which end up in the blood circulation. Something related to that I wanted to just briefly touch on is the titer. When people talk about what is the titer of such and such, they're talking about what's the concentration of antibodies. What is your titer to tuberculosis? What is the concentration of antibodies that you have against tuberculosis? Or what is the level of antibodies that you have against measles? The higher the level, obviously, would indicate that you've had some exposure, either natural or artificial. You either had the disease or you had vaccination against the disease. You have generated those antibodies, and it's at a high enough level to protect you. One thing we do find with the whole vaccinations, immunization records, is that over time, sometimes these titers tend to decrease. And that's why oftentimes in healthcare especially, they will periodically monitor the titer, which is monitoring the concentration of antibodies against a specific organism. They may want you to get a booster. A booster is going to trigger that memory response so that you have increased production of the antibodies to get the levels back up high enough. It will provide enough adequate protection against you should you be exposed to it. So typically, as the antibody increases, obviously the titer is going to increase. If the infection decreases, the titer will then decline. This is a slide I really wanted to show just to emphasize to you. You have with the adaptive immune system these two components, the humoral, which is or antibody mediated, this involves the B cells, and you have the cell mediated, which involves the T cells. This shows the interaction between the two. You have initially your antigen presenting cell, which is a macrophage, is a dendritic cell that engulfed, it ate up basically the foreign substance. How did it know it was a foreign substance? Because it did not have your MHC on it, your major histocompatibility complex. It didn't see it. It said, oh, this is not part of you, so it ate it. What the T cells do, then they will activate once the antigen presenting cell, which ate the foreign substance, it essentially will show this foreign substance on the surface of its cell in combination with the MHC. And it's basically flagging it or putting a red light. Here it is. This is what you need to look for. We have an invader. The T cells will become activated. And the helper T cells They will produce cytokinins. This will activate macrophages and attract them to the site so that you can have further, it's basically calling in the help, the backup to help fight this infection, because they will be able to recognize anything foreign and engulf it. The helper T cells, it doesn't show you, they're going to actually also help. You have two different types of helper T cells. They're going to help activate the cytotoxic T cells. Some of these are going to then be going out, searching for that foreign cell that has, anytime it sees that foreign antigen, it will attack it and it will destroy it. Some of the cytotoxic T cells will also become memory cells, so any future infection, it will be able to target those invading cells as well. The helper cells, meanwhile, they will also activate your B cells. Your B cells activate the plasma cells, which activate the antibodies. When the antibodies bind to the foreign antigens, it flags them, and it will also attract the cytotoxic T cells. So these two sides have to work together. So the T cells typically are activated first. You get the activation of the cytotoxic cells, the helper T cells. You also have regulatory T cells, which are keeping these two components in check. The helper T cells activate the B cells. If this process right here did not happen, you would never end up with the antibody production, and that certainly is going to have a huge effect on your ability to fight the infection. The helper T cells, there's two different types. One of them is called the CD4 cells. The CD4 cells are involved with helping to activate the B cells. Some of them also will work to help activate and increase the numbers of the cytotoxic T cells. Think about this for a moment. If you wipe out the T cells, you do not have an immune response. And that foreign substance can just really enhance its invasion of you. The HIV virus, which is the causative agent of AIDS. One thing we have found, that one aspect that it has, is that it will inhibit the CD4 cells. It interferes with it, decreases them. Basically, it's wiping out your helper T cells. If you wipe out the helper T cells, you're never going to have them affecting the cytotoxic T cells, because as you can see here, it goes back and activates more T cells, which then activates the cytotoxic T cells back here. So if you wipe these guys out, you wipe this out right here, which ultimately means you wipe out any stimulation of your cytotoxic T cells. None of this will happen. Infected cells will not be destroyed. Memory cells will not be made. If you wipe out the helper T cells, you also cut that link between cell-mediated and humoral immune system, which means you never get antibody production. Very bad for you, very good in this particular case for the HIV virus. It's a way of knocking out the immune system, so you're knocking out the cells that are trying to kill you. It's very effective, very good in terms of the virus, very bad for you. But I hope that this short little presentation would help to explain overall the adaptive immune system and how the components have to work together.